then I'll open this up. Hopefully everyone has access to this. The Colab notebook should work in your web browser. And in this notebook, much like the machine learning part, we have the kind of the same problem, where we're predicting gene function using deep learning now. So what I've done basically is taken the notebook we worked on in the machine learning lecture, but now instead of applying models from scikit-learn, we're applying different deep learning models, and we'll see how they perform, if they're able to do any better or worse than what we did with scikit-learn. So again, to remind you of what we did, so we went to Harmonizome and we got the diseases data set, which is curated from literature and gives these associations between these disease codes and particular proteins. This is all from literature and only encompasses 2,000 genes. And we're going to use these as labels for our machine learning problem. We then get from Geo, which has various experiments where a gene was knocked out, overexpressed, or mutated. And when we load that in, we can see that we have 22,000 rows, different genes that have these relationships to these other genes that were knocked down or knocked out. And ultimately, this represents this rather rich feature matrix, given that their relationship to all of these, within all of these different experiments to other genes, should, sorry, I lost my place. Their relationship to all of these genes should give us some sense of the function of that gene, based on like how it's affected by its, in these particular experiments. And then so we looked at diseases, and then we found as a topic to predict just this generic eye diseases. And once we selected those labels, we had our kind of true known genes associated with eye disease. And we prepared our data to get, our X and Y here, where we have, this 739 relationships in geo experiments to all 20,000 genes, all 22,000 genes here. Yeah, so I will move onward. So finally, we still need to remember to keep this test data set separate. So we need to be able to benchmark it later on. So we take out 20% of the genes and we save it, or rather 20% of the data, and we save it, and don't show the model that, so that we can validate how good it is later on. We find the class distribution, since this is imbalanced, and we want to make sure the model does not become biased because of this. And we have our scikit-learn based validation function from before that benchmarks how good the classifier is doing. So um, I show this first model. So what we're using is TensorFlow again, and TensorFlow has something called Keras as a submodule, and it allows you to, at a very high level, express what you need out of your machine learning model, with code, and this ReLU activation layer that I talked about is defined like this. It's just max 0x, which means that if the output is ever less than 0, it will just be 0. But if the output is more than 0, we keep it. We don't adjust it in any other way. Even though this is kind of linear when it's greater than 0, it's still considered a non-linear function. And we saw in the playground that even though it's linear, it can fit to non-linear outputs. And the way it does this, because it has this 0 part, it can kind of chop off the left side, and it can stack these lines together in a way that's not unlike how the decision trees are functioning. It's able to draw these lines around things, and that really is what the ReLU is enabling the network to do. But on the output, we're going to have this sigmoid because we have a binary classification problem. Sigmoid, again, is defined like this, where you have this exponential in the denominator, and the bounds of this function are between 0 and 1. And the loss function that we apply, the binary cross entropy, is going to turn this into a probability distribution. So we're really learning like the probability that this data is a 1 or a 0. That's what the loss function will end up pushing this value to be. So to make this feed-forward neural network, we define this sequential type. So sequential makes it pretty easy to just stack the different layers horizontally, if you recall that image. So we have the input layer, which is of shape the data that we had. So our data is here, and we're actually using this shape. So this is 739 different columns. So the shape here will be 739 in the input. And we then just stack 3 dense layers. So remember, I noted that a singular dense layer will just take everything from this layer, multiply it by the 64 different weights on this layer, add it all up, and then apply ReLU, and then you have the output to this layer. Then we do the same thing with this layer, and we feed it into the next layer. So we keep doing this, down the line until we get to the bottom, where all 64 of these layers go into a ReLU, and all 64 of these layers are used to predict 0 or 1 at the bottom. So that's it. It's like pretty simple, right? We put in the input. We have all these, this 64 is arbitrary, and you can tune these as such until you get the classifier. The next step is to compile. So when we do a model.compile, we have to specify what is the optimizer we're using. So this is what's doing gradient descent for us and going to tune these weights, right? So we have 64 here. So, but the 64 doesn't mean there's 64 weights. There's going to be this many. It's going to be a matrix that is of shape this one by this one, and again 64 by 64, then 64 by 64, then 64 by 1. So there's a lot of weights there, a lot of parameters, and the atom's job is to, tweak all of these weights until the output is what we want. The binary cross entropy loss helps us do this binary classification. And then we also have these metrics. So we're doing a receiver operating characteristic, ROC, and we're finding the area under the curve, and we're also finding the recall. And this is from the machine learning lecture, where we learned that for classification problems, these are two metrics that are very helpful to use. So once we compile it, we can now fit it. And so what we do is we fit it to the training data. We test it on the testing data that it never used. And so here's how we do that. So we run this model.fit that we've compiled. We give it the X and Y training data. Then there's some other parameters we have here. So we have shuffle. This just makes sure that for each epoch, it won't just use the data one by one, one after another. We'll actually shuffle them around a little bit. And this helps to impart some stochasticity. Remember, we talked about if the optimizer gets stuck. So even just like changing the orientation, like the ordering of the training data you give it, you can help get past some of these valleys. We specify the class weight, which ensures that we don't since we're seeing so many false labels and so few true labels, our model will become very biased towards false labels. So the class weights helps us counter that, so we don't become biased. And Keras actually lets you specify the test data straight in this fit function, and then it will compute metrics on the test data as we go along. The epochs say, okay, we're going to give the data to the model 200 times. So this model will get all of the data and it will run through 200 different times. And the batch size tells it, okay, so this is taking the shape of it and dividing by 5. So that means we're going to have a fifth of all of the data in each batch. And a batch is kind of just a implementation detail. So basically, we could train one by one, but this would take a long time. So the idea is, let's instead of training one by one, let's train in parallel an entire batch of them and give it to the GPU, because GPUs can do these matrix operations in parallel rather rapidly. So that's what batch size is about. When you do this, you end up using a lot more memory. So basically, you can make the batch size as big as you have memory, and it will speed up training substantially. But batch size can have negative implications on your actual learning, because the error signal gets back so late. But so, okay, we trained it, and then we look at the validation and the actual, like the training and validation loss. And this is the plots that we have. So the blue is the what we're training on, and the yellow is what we're validating. And you can see that within about 50 epochs, we're pretty much converged to what we're going to get. And here we can see the problem of overfitting. So our model, even though the regular loss is very low, the validation loss starts getting worse. In the AUC, we can see that by 50, we have a perfect AUC on the training data, but on the testing data, we're at random, 0.5. And for recall and precision, we can a similar thing where the training data has perfect recall and precision. We've essentially memorized everything there, but in fact, the validation set never learns anything. It stays at 0. We never recalled anything. We just always get it wrong. So that's not good. So let's see if we can do it a little better. We can also look at just these plots that we had in the scikit-learn case, and we see the same thing. So this is now evaluated after we've trained. So here we're seeing kind of just these metrics during training, and at the end we're then looking at these like this is what we got out of it. This is our fit model. So you can see that we basically are perfect besides this 6 on the test on the training data, but on the test data we're just wrong. We have some things that we predict, but they're always wrong, and in fact we may be doing worse than random. So what can we do to fix this? So let's see. So in the next step, we still have these 364 dense layers, but now I'm introducing a few additional concepts. So one of them is batch normalization. So batch normalization is useful for making sure that when we go through, we don't get like these how do you say this? So when you pass you want to make sure that the data within that batch is normalized, and you don't have this problem called like center mean shift. So it's, let's see, do I write it here? So I wrote batch normalization helps to stabilize training by reducing the weight distribution shifting from zero mean, discouraging the learned parameters from exploding indefinitely, causing numerical instability. So the idea really is that your weights can keep getting bigger and bigger. So if they're not getting better, they just keep getting bigger and bigger. And this can be a problem, obviously, because if they get too big, you get out of the range of a floating point, and now you have numerical stability problems. So what batch normalization ends up doing is every time you do a pass, it's normalizing all the data in the batch with respect to that batch, so that the weights don't end up exploding. They stay kind of normally distributed around 0, and this is very helpful. Another thing that we add is dropout, and this will randomly set some of the weights to 0 during training, and this has like very good effects because basically it's, even though we have so many parameters, now some of them are not available. So our model starts behaving a little bit more like an ensemble. some of the paths in the network are redundant with others. And so any like noise kind of gets averaged out by the ensemble that we end up with. So it does end up like requiring that we have to train it for longer, but it has the nice benefit that it does reduce overfitting. So with these two tricks, batch normalization and some dropout, let's see how we do. So now I train and we can see that while our loss is very low, our validation loss is much higher, but we do see a good sign that for the entire duration of training, it continues to get better. Even though it's not great, it is getting better. And we see the same thing when we look at the ROC and precision recall. We can see that while our training ROC kind of goes from random and improves progressively, and our validation ROC turns out was like good at the beginning, and this is really just a artifact probably of the smaller size. But we can see that the training is improving and the validation, it has kind of plateaued and it's starting to go down, which isn't a great sign, but it's certainly better than what we had before. And there's some evidence that we might be able to train this for longer and do a little bit better. And that's what I was mentioning with dropout, because we are kind of deleting all this stuff. We need all these redundant paths, and it takes a lot longer to train. But you end up with a more generalized model after you do train. And for precision recall, we can see we're pretty low in general on this. And I think this is just due to the fact that we have a pretty imbalanced data set and we haven't trained for long enough. If we take this particular model and we like benchmark it with our previous scikit-learn benchmarking function, we can see that the training ROC looks great, and the testing ROC looks pretty good as well. In fact, it's competitive with what we had for scikit-learn and XGBoost. In fact, I think it may be doing a little bit better than XGBoost here, which is a great sign. So again, once we've actually trained these models, we can then get outputs from them. So to do this, you want to train the model after you know that it can generalize to an extent. You want to train the model on all the data, which is what we do here. And because we don't have validation, we can't get that curve, but we can see that the loss function continues to decrease. And again, there's evidence that we probably should train this for longer to get better results, since this is not, it's just going down, and probably will keep going down if we train it for longer. But we can take our model, we can give it the X to create prediction probabilities, and we can again make this table where we have, this is the gene that was predicted, and we can take out the ones that are 0 to find novel gene associations. And again, these predictions then suggest that, at least according to this particular model, which is not a spectacular model, that these genes may in fact be implicated in some form of eye disease. Yeah, and that's really all I have for today. But if there's any questions, definitely let me know, and I'd love to elaborate further.
I have a question. So you just mentioned that like this model isn't like a particularly great model. So what next steps would you take in order to make it a better model?
Great question. So this is kind of the tools of the trade, if you will, and a lot of it is really just, it's finicky. So it's a lot of tuning these things. So on one hand, um, these dropouts and the actual numbers here. So I have 64 now, but we can use bigger layers. We can use smaller layers. Um, we can use different amounts of dropouts. So dropout 80% is pretty high. We can make it less, and it would speed up training, and potentially we could get better results. We can have more, so we only have 364, but with all of these things, there's some diminishing returns, right? So even if we have many, many parameters, now it's slower to train and potentially can overfit a lot easier. So in some ways, you do want to keep it smaller. We might try getting rid of one of these to just have one. But in a lot of ways, it's like tweaking these different things. And there are some approaches that I didn't cover, but it's this whole field of so-called meta learning, where you are kind of tuning these high-level architectures based on these hyperparameters, and so you use machine learning to make your machine learning better. um, but that's kind of what it is. I think the biggest thing, it's just like, can you get something that behaves mostly well in a short period of time? And but that would be my next step. It would, clearly dropout and batch normalization helped a lot. So it might just be a matter of tweaking these dense layers and the number of them. Um, but it's also, you know, might just have to be more specific, right? So if you remember at the onset of this whole thing, we're trying to predict eye disease, which is a very broad concept. And, this could be part of the reason why we don't get spectacular results. If we have something that's a little more clear cut, we'd probably end up getting a better model as well. So these are just some ideas, but the thing to always remember is that these models are very powerful and they can very easily learn nonsense. There are very famous examples. For instance, this study where they were predicting breast cancer from images, and they basically learned that if there's a ruler in the image, the person is more likely to have breast cancer. Um, this was not a desirable outcome, but it was a fact because the data that they gave it, if it was a tumor, they had like a ruler in there. So this is definitely something to be aware of, that there can be these types of issues. But I hope my tangent was not too much, and I hope that answered your question. It's a matter of tuning these things. This is an NLP architecture, but like I showed a bunch of other architectures. You could presumably apply some of those architectures as well if there's a way to build an embedding based on how genes relate to one another using a different data source, right? That could apply to this situation as well, right? Maybe you look at how do genes relate to one another and do a word to vec style embedding for the genes, and then now okay, these genes are related to one another, and that type of embedding might be a much better input than like what we have here. So there's so many ways we could go about improving this. Um, but hopefully that gives you some ideas.